invitation to talk today. Is this too loud as I'm speaking, or is that all right? I invite everyone as we go along here, if you have any questions, it's fine to interrupt me and ask questions as I go along. You don't have to wait till the end, so don't be shy. This is sort of the title of the talk. I'm going to cover a lot of area, but it really relates to the importance of this simple molecule called nitric oxide or nitrogen monoxide and resistance to malaria. And this is something that we've been studying for a long time with my collaborators, Dr. Makambo in Tanzania, Dr. Yo and Dr. Anstey in Australia, Dr. Granger in Utah, and several others. Now, I know that this is the global health group, so you've probably tuned into this. I'm going to cover a little bit of background about malaria to get everyone at the same level. And I'll be speaking to some people about things that you already know, but other people may be new to you. Now, most of the problems from malaria, the deaths and the disabilities, come from severe malaria. And that's really related to cerebral malaria, where patients develop coma, grand mal seizures, and frequently death. And then they can also have severe anemia, and compared to what we deal with here in the United States, where we get all upset about anemia with a hemoglobin of maybe 9 or 8 or something, and the definition of severe anemia there is a hemoglobin of less than 5, where a normal hemoglobin might be 12 to 14. So it's really severe anemia. Anemic enough to where patients die because they don't have enough red blood cells, they don't have enough hemoglobin. Severe anemia. There are a lot of cases of malaria, 1.5 to 2 million cases per year. It's difficult to get an accurate count, but that is fairly accurate. And importantly, there are 600, 800, and up number of deaths per year. If you go through the math and calculate that, that means maybe 70 to 90 people worldwide are dying from malaria every day or every hour. And most of the deaths tragically occur in children less than 5 years old and in pregnant women because of lack of immunity and some immunosuppression. And despite our best treatments that we have for malaria, something like 15 to 17% of people who come in and get our best treatments go ahead and die. So we need some new treatments. In addition to the deaths and disabilities, in these populations in the tropical areas, when people have malaria, it leads to a great disability in this population. They're not well enough to work. They are just so tired and anemic, et cetera, that it really decreases the productivity of the whole country. And that's been documented very well. And importantly, there's not a malaria vaccine available. We sometimes take for granted how vaccines are so important to us all over the world. And I just list several vaccines that are available. Many of them are outstanding. Some of them are not perfect, but if you just think about a few of them here, like tetanus vaccine, measles, mumps, polio, herpes zoster, etc., strep pneumonia. These are really good, and some of them are not fully effective, but many of them are offer greater than 95% protection. However, there is a great need for other vaccines, and leading the list are really HIV, malaria, and tuberculosis. There are a lot of people working on this worldwide. At Duke, as you know, here in the Human Vaccine Institute, there's a large thrust trying to develop HIV vaccines, but also other vaccines. So if we had a vaccine for malaria, we'd be in good shape. Now, within the last year, there have been several high-profile publications making you think that we're really just about there with that malaria vaccine. But I'd like to say that we're really not just about there if you really look at the data in this. And in this study, for example, that was published in the late 2012 in the New England Journal of Medicine with a recombinant protein-type vaccine, if it was measured at 12 months after giving a series of vaccines, only about 30% to 35% of people were protected against any kind of disease, including severe disease. So that's not going to be good enough. Actually, the FDA is close to approving this vaccine. The other thing that got a lot of play in the press was a vaccine in which Dr. Steve Hoffman and his associates have been working for years on, where they take the mosquitoes. Here's a picture of that here, where we've got sort of a mosquito on a dissecting tray with a needle. And in the life cycle of the mosquito with the malaria, life cycle of malaria in the mosquito, it eventually goes to the salivary glands. And so these workers, in an industrial fashion, in a sterile fashion, take the sporozoites out of the salivary glands and purify those, and then do some work with them, and then eventually put those in the and make a drug out of them for a vaccine. And in this particular experiment where they used these purified vaccines or purified sporozoites in this vaccine, they then gave injections of this to try to immunize people. And after 5 injections intravenously, now think about this worldwide with all these people and children that you're trying to immunize, 3 weeks after the last injection, so they analyzed it at this early time point, they did show 100% protection against the subsequent challenge. So it sounds like a good vaccine. But it was extremely high dose with the sporozoites. And they did, it's a beautiful paper where they do a lot of science with it, like looking at T cells and vector T cells, antibody levels, et cetera. So they're getting there. My point here is we're still a very long way away from a malaria vaccine. So it'd be good to have some better treatment as far as drugs and prevention. Our work in malaria research started here at Duke in 1994. And at that time, people in this picture, so myself, Dr. Anstey, and Dr. Granger were here. So I and Dr. Granger were on the faculty. Dr. Granger in infectious disease. I'm in hematology oncology. And then Dr. Anstey was an infectious disease fellow. He and his wife spent a year in Tanzania collecting samples from children with malaria and doing studies to try to understand the importance of nitric oxide. And then over the years, we've continued this collaboration. Dr. Anstey's now back in his Australia, where he's from. Dr. Granger's back in Utah. And then importantly, Dr. Esther Makambo has been central to all of our studies, where she's been the principal investigator and leader of the studies in Dar es Salaam. So not Moshi that you hear a lot about with KCMC, but it's in Dar es Salaam, Tanzania, the capital of Tanzania. Now, you may remember, and I'll go ahead and go through this, although I know everybody knows the cycle of malaria. We can have infection with the mosquito, where you get bitten by a mosquito, and then the sporozoites enter the skin. They traverse the skin. They get into the bloodstream relatively rapidly. They go to the liver.
Sorry, I think we're going to have to unplug your hotel microphone.
Maybe if I just move it down some. Is that better? I guess there is an echo. Maybe it was too close. Anyway, it gets into the liver. The sporozoites infect the hepatocytes, and then they multiply and divide in the liver cells, and then it ruptures out. And then the merozoites are the next infective form that come out of the liver, and then they invade red blood cells and set up infection in the red blood cells. And then after a few days, they go ahead and eventually rupture and releasing more infected merozoites. And if a mosquito happens to come along and bite that person again when they have malaria, they'll pick up an infected red cell, and they're able to start the cycle again, usually in another person. So this is kind of the cycle. Now, each step here, it's very important that you understand that each time the red cell invades an hepatocyte, or rather the malaria parasite invades a red cell, there is this adhesion phenomenon at the cell membrane. Likewise, in the placenta and the endothelium per se, the infected red cells can interact with endothelial cells, and this is actually a receptor-mediated interaction where there are known specific receptors. So that might give us a target to look at for interrupting this infectious cycle and the pathology here. These are some short movies that I got from the Walter and Eliza Hall Institute in Australia. I would recommend you go to this website. There's some movies that are much more sophisticated than these, which are just terrific, looking at the life cycle of malaria in us, the humans, but also in the mosquitoes. And as we see here, red cells sort of floating through the blood, and then let's look at this as a merozoite, a malaria form that will sort of touch a red cell, and then through a receptor-mediated interaction, then actively invade that red cell. And once it gets to the red cell, it starts multiplying and dividing. The source for nutrition is hemoglobin, so it eats the hemoglobin to digest it. It also causes formation of methemoglobin, this sort of darker maroon color. In the flowing blood, these infected red cells become adhesive, and they actually stick to endothelial cells. And this endothelial adhesion of a parasitized red cell can just be one red cell, but frequently it's multiple red cells, and red cells with platelets and red cells with white cells. It eventually ruptured, releasing more merozoites. And it's thought at that point where it ruptures and releases those malaria products into the circulation is when people have fever. They're so-called malaria toxins released, which are glycophosphoridol and inositol type compounds, the fever and rigor that people have. So there are a lot of red cell interactions here. And these interactions can be mediated by parasite proteins on the red cell membrane. Once the red cell is infected, the parasite encodes proteins which are inserted into the membrane of the red cell. And so those are the ones interacting with our cells, like endothelial cells and leukocytes. And with this going on, we can have parasitized red cells reacting with other parasitized red cells, or parasitized red cells interacting with a normal red cell, and then these interacting with platelets and other leukocytes. But most important for the pathophysiology is the interaction of the parasitized red cell with endothelial cells, the lining of the blood vessels. We think that that's very central to all of the pathophysiology of malaria. These are mediated by specific receptors. These receptors are used in all types of biology, in cancer biology and normal physiology, like vascular cell adhesion molecule 1, intracellular adhesion molecule 1, et cetera, et cetera. If you think about this and relate it to the pathology, let's think about malaria and pregnancy, a very severe problem. It's known now, work actually by Dr. Patrick Duffy, a person who was here a few years ago speaking, and a person who actually graduated from Duke Medical School and worked with Dr. John Perfect when he was a medical student. He discovered that in the placenta vasculature, chondroitin sulfate A and some other molecules comparable to that serve as a receptor for the parasitized red cells. And they are looking at ways to intervene and interrupt this problem in the placenta that causes early delivery, abortion, and small birth weight children. In blood vessels in the brain that are involved in the mediation of cerebral malaria, and in blood vessels all over the body, like just the capillaries, there's a whole series of receptors that have been identified that serve as specific receptors for that parasitized red cell binding to the endothelium, like thrombospondin, ICAM1, et cetera, et cetera. Now, I'm going to make the jump to nitric oxide, and the transition about nitric oxide, based on what I just talked about, things adhering to endothelium, is that nitric oxide influences the adhesion of red cells, but also platelets and other cells, to endothelium. So I've worked with nitric oxide for a long time, and I look at it as Randy was surveying the different areas where I work. It seems like I'm all over, and I am, but it's sort of pulled together because a lot of the pathology and physiology in blood cell function, in arthritis, inflammation, in obstetrics, and infertility is actually mediated by nitric oxide. So we use the same techniques in a lot of diseases. Nitric oxide is a reactive free radical. It's produced in our bodies normally. There's several people who work in this area. Nobel Prizes have been awarded for research in this area. It mediates host resistance to infectious organisms, including parasites. And it's synthesized by one of three separate genes, NOS1, NOS2, and NOS3. There's been a lot of work in malaria with nitric oxide. In the late 1900s, investigators showed that hepatocytes making nitric oxide, remember there's this hepatocyte cycle, part in the cycle of the malaria parasite, and also monocytes in vitro will reduce the viability of parasites that are in red cells. And probably the unique compound that does that is a nitrosodiol. It's sort of a special nitric oxide species. In the 1900s, there were a group of investigators that felt like nitric oxide was actually what was causing people to be sick. It was known that nitric oxide is important for neurotransmission in the brain. It's a signal transmitter. And also, nitric oxide could cause toxicity to the brain. And so they then deduced that, well, maybe these people are got activated cells all over their body. They're making a lot of nitric oxide that causes convulsions and coma and death and it's the cause of cerebral malaria. That was the going thought. And then when we started our experiments with Dr. Granger and Dr. Anstey and Dr. Mike Combo, we were trying to either prove or disprove that. As I'll tell you today, we sort of disproved it. And then we showed that nitric oxide, instead of being a mediator of toxicity, was actually protective. And the anti-disease effects of nitric oxide, we think, are related to the ability to decrease cytokine formation, like these protein mediators that cause disease, like tumor necrosis factor, interleukin-1, to decrease expression of these cell adhesion molecules. So if you have an endothelial cell expressing all of these cell adhesion molecules and you treat it with nitric oxide, those cell adhesion molecules go away. So the parasitized red cells can no longer bind to the endothelium. It's sort of a simplification, but that's a good way to remember it. And also in our bodies, nitric oxide, one of the main functions is to cause vasodilation. It keeps the blood vessels dilated by relaxing smooth muscle. Nitroglycerin, which is used in cardiology, been around for a long, long time, mediates effect by serving as a prodrug for nitric oxide. It releases NO, dilates blood vessels. And these anti-disease effects, we think, are so important in what's going on in malaria. So we've been working hard to understand that. Now, nitric oxide is synthesized by this specific enzyme called nitric oxide synthase, as it converts arginine to nitric oxide and another simple amino acid, citrulline. And if you think about just general basic biology and chemistry, we've got DNA with the gene encoding an RNA. So this is transcribed to the RNA. The RNA is translated into a protein, and then that protein does something. Here, that protein converts arginine to citrulline and nitric oxide. Now, in this whole sequence, it's much more complicated than that first slide, in that there are several things that influence this. First of all, in the gene, there can be certain things that influence the ability to transcribe that particular RNA. And we've discovered, so I'm sort of going to go through this and tell you some of the things we've looked at and found. We've discovered that there are certain single nucleotide polymorphisms, or like mutations, variances in the gene that will cause different levels of transcription in people. Some people are really good at making this message and making nitric oxide synthase. Some people don't do it at all. We've also, and then not just us, but other researchers for sure, have shown that there's a group of molecules that will influence transcription, and these are like tumor growth factor beta, TGF-beta, IL-10, steroids like prednisone, hydrocortisone. They will inhibit this transcription. Other things enhance it. So these are things that sort of turn on immunity, like gamma interferon, alpha interferon, lipopolysaccharide, or endotoxin, TNF, et cetera. And then down here, and I won't spend a lot of time with all of these, but down here, once you have this enzyme present, it requires a whole set of cofactors. And if those cofactors aren't there, that enzyme does not work. And one of the most important is tetrahydrobiopterin, that I will talk about later. If you are low on some of these, like if you're low with this biopterin, the protein doesn't work. If you don't have the substrate, if you're low on arginine, then you can't make nitric oxide. There's no arginine that can be converted to nitric oxide. And then interestingly, in some work we've recently published, and I'll show some of the data, in our bodies there are analogs of these arginines that are made that sort of act like drugs that we would use in clinical medicine if you had them. They actually inhibit the enzyme, and they're made in our bodies, and I'll talk to you about that. And then finally, if you make nitric oxide and have it present, there are things around that will actually blunt its effect. They act as scavengers, so they sort of bind it or get it out of the way rapidly. And the main one that's important in malaria and also in sickle cell disease, where you're rupturing red cells. So if you rupture a red cell, hemoglobin comes out, and it's free floating in the plasma. And so that hemoglobin, it's really the heme in the hemoglobin, will bind nitric oxide as one of its effects. So there are different steps, multiple steps in this whole pathway where things can get sort of screwed up if you don't have enough nitric oxide available to mediate its biological effects. And this is what we looked at.
So Brice, why do oxygen and heme appear on both sides of that slide as positive and negative mediators?
Well, oxygen is absolutely necessary for the synthesis of nitric oxide in this reaction of nitric oxide synthase. So there in right amounts it will. If there's too much oxygen, and in different forms, at least superoxide, etc., it can be inhibitory because it will oxidize the heme and the NOS protein, and it will also bind or quench the nitric oxide itself. That's a common question. Now this next slide is sort of, sit up, look at this slide, because this is all I'm going to tell you the rest of the talk, but I want to make the points here. These are things that we've found that in this nitric oxide arginine pathway that are protective against clinical malaria. This is our evidence. First of all, we've shown that there's low overall nitric oxide production. So in patients, not just cells in their test tube, but in the patients, we show low nitric oxide production. Also, in special studies, we've shown that there's low messenger RNA for this nitric oxide synthase gene. There's low protein, and there's low enzymatic activity. So all of these are not making enough NO here. And there are certain NOS promoter polymorphisms that modulate messenger RNA levels. In one situation, we actually found one where the people would make a little bit more nitric oxide, and those people were protected against severe malaria. There's low nitric oxide synthase cofactor in work we haven't published yet. There are definitely reduced amounts of this fully reduced biopterin, so-called tetrahydrobiopterin. That's that critical cofactor that's got to be there. But for some reason, and I'll talk about that, there's not enough being produced in the bodies. And then importantly, we found that patients with clinical malaria, either uncomplicated or severe malaria, have extremely low levels of arginine. The normal levels are around 100 micromolar. These people have around 35 to 40 micromolar, levels that just aren't enough to make nitric oxide. And then in addition, people have increased levels of an enzyme called arginase that sort of gobbles up or destroys arginine, another way to deplete arginine. And as I mentioned, there are these endogenous inhibitors of nitric oxide synthase that will block nitric oxide formation. I'll expand on that in a minute. And then finally, we've measured plasma-free hemoglobin in patients with malaria. And as part of this hemolytic pathway, it's like we lost our picture here. In the hemolytic pathway, there's hemoglobin that's released into the plasma, and then this plasma hemoglobin quenches nitric oxide. In the early studies we published in 1996, the first work coming from Dar es Salaam, we showed in a Western blot, this is where you're able to analyze protein extracted from the cells, that patients, the healthy control patients, these are people who just take normal controls, these were children who we documented had no malaria, they would express some of this nitric oxide synthase. It's this little bar across here that's stained in a special way. There are some people that we would find in our healthy control group, they weren't sick at all, but when we looked carefully in their blood, they had parasitemia. We found parasites, and this is known to happen. Some people will have asymptomatic parasitemia. They'll find the parasites, but they don't have fever, they're not sick. They actually had very high levels of the nitric oxide synthase. In the most sick people, most sick children, cerebral malaria, they really had no nitric oxide synthase. So once again, no protein. And here's sort of the statistics from that down here. And in general, there was sort of an inverse correlation of systemic nitric oxide production with malaria disease severity. If you had high levels, it was associated with absence of any clinical disease, either with the healthy controls or the asymptomatic parasitemias. The square here is the amount of the NOS protein, and the diamond is the amount of nitric oxide that we measure. Likewise, or contrary, I guess, low systemic nitric oxide production was associated with clinical disease, like uncomplicated malaria. People come in with fever and chills, need to be hospitalized, or cerebral malaria. And when we quantified this from the blot, this is done some time ago, we showed that the amount of protein based on the immunoblot was highest in healthy. We didn't do the asymptomatic parasitemia patients here. Lower in uncomplicated, and most low, the lowest, in the severe malaria patients. Likewise, if we did the NOS activity, we have radioactive arginine. We can measure conversion of arginine to radioactive citrulline, which is sort of get nitric oxide. This was also reduced in a comparable fashion. And then when we did the messenger RNA by real-time quantitative PCR, we showed once again that those patients who had malaria had very low levels of the NOS2. So just in summary again, there's an inverse relationship to malaria disease severity in Tanzania, and we also did this in Gabon with some researchers there, and in Indonesia and Papua. Adults, we did it both in children and adults, and showed the same thing. High NOS associated with no disease or mild disease, and low NOS associated with severe disease. I mentioned the promoter. I'm not going to take time to go through all of this. This was published in 2002, where we demonstrated this specific NOS2 promoter, that the single nucleotide polymorphism that was associated with slight increased production of nitric oxide and provided protection. We've documented several other promoter polymorphisms and certain haplotypes that are associated with either increased or decreased nitric oxide and change in susceptibility or manifestation of disease. The other thing we published, I guess this was 2003, is, as I mentioned, that children with malaria, and we've now shown this in adults too, have very low levels of arginine, the substrate or precursor of nitric oxide. And this shows some of that data. In healthy controlled children, as you see here, the individual data, and then the median, I think, maybe it was the mean, around 100 to 120, uncomplicated malaria lower, severe malaria or cerebral malaria even lower. And then the different colors, the black and the white here, I won't go through the details, but we found that those with the lowest arginine were the ones that were most apt to die of their cerebral malaria. So it was actually, the level was predictive of death. Now, I've talked a lot about endothelial dysfunction, and we have done a lot of studies about endothelium. This work was actually led by Dr. Sen Yo, who was at one time an infectious disease fellow in Utah with Dr. Granger, and then eventually went to Darwin, Australia, Northern Australia, and has helped with Dr. Anstey to head up all of the studies in Indonesian children and adults. And they did extremely important, detailed, and I think really exquisite studies looking at endothelial dysfunction in malaria. Now, just to back up a little bit, I mentioned activated or sort of modified endothelial cells have increased expression of these cell adhesion molecules. So they're more apt to bind something. They're actually more apt to bind white cells, but also red cells, and especially parasitized red cells that have malaria in them. So these parasitized red cells do bind to endothelial cells, and then once a parasitized red cell binds to the endothelial cell, it increases the expression of these cell adhesion molecules even more. So you can see now you're going to get into sort of this cycle of bind more, express more, bind more, and you get this cycle of increasing endothelial cell alteration in malaria. And the important thing here is that nitric oxide influences both the expression and the adhesion. So if you look at this as a cycle, this is a slide I think Dr. Anstey and Dr. Sin prepared and gave to me. Let's say you have a situation with a lot of pro-inflammatory cytokines like IL-1, TNF, and then you're getting increased endothelial cell adhesion molecules, and then these parasitized red cells bind to them. This is a photomicrograph of a vessel from a brain of a child that died from another paper, not ours, another one. And these are parasitized red cells clumping up and binding. And part of the pathophysiology is there's so much clumping in the blood vessels that it can actually inhibit flow. So there's an anatomic inhibition of flow because they're getting plugged up. And then once they're plugged up and binding, you get more inflammatory cytokines, so around and around you go. And then nitric oxide really blocks every one of these steps. So if you've got enough nitric oxide, you can help interrupt this bad problem here. So how was it that we measured endothelial dysfunction. Once again, it was Dr. Yo and Dr. Anstey who initially set this up and made it work. The cardiologists have worked in this area much, and much of the work in nitric oxide and blood vessels is done by cardiologists, and they're outstanding in that area. We were able to measure what's called reactive hyperemia peripheral artery tonometry. So what happens is we take a blood pressure cuff, put it around the arm, elevate it to a point above the systolic blood pressure so you don't get any blood flow to the arm, and you leave it there for 5 minutes, and people can tolerate that. It's not dangerous. And then you let it go, and once you let it go, the blood flows through very rapidly, just gushes through. And so you get a lot of force over the endothelium, and probably actually partly mechanical force. It increases nitric oxide liberation from endothelial cells, and then that NO will give you a extra dilation of the blood vessels, and I'll demonstrate that. So nitric oxide is responsible for more than 50%, some people say a lot more, of this vasodilation. So it's called peripheral artery tonometry post-arterial occlusion. And work related to that was published in 2007 in Journal of Experimental Medicine. I'll review part of that. This is a picture from Indonesia, where we have a patient. This may actually have been a normal control, where there's a blood pressure cuff here and a blood pressure cuff here. There's a little finger probe, kind of looks like one of these oxygen things you get put on your fingers to measure O2 saturation. But this was to measure pulse waves. And it's hooked up to a machine and then a computer. And once the blood pressure cuff is blown up, you can sense this in the pulsing in the finger artery pulses. And that's demonstrated here, taken from this paper by some cardiologists. So here we have one arm in which, this is a typical response. We have the pulses. Each little thing is a beat of the heart with an arterial pulse. And then the blood pressure cuff is blown up, so you stop the blood flow. And then you release it. And then you start getting the pulse waves again. And notice the height of this is much higher than that. So there is this vasodilation, and so you get increased blood flow. Down here is the control arm, where you don't put a tourniquet on. You don't blow the tourniquet up. And so the blood pressure is not interrupted, and this height of this is the same as before. This is actually from a patient with severe atherosclerosis and hypercholesterolemia. You get a defective post-occlusion vasodilation there too. And this was measured in patients, the healthies, and then those with moderately severe malaria and severe malaria. And the finding was that in patients who had severe malaria, and this was all in adults in Papua, Indonesia, there was a statistically significant decrease in this so-called reactive hyperemia peripheral artery tonometry value. And in these patients, we also measured arginine levels. So once again, as I mentioned, they have low arginine, and we administered arginine intravenously. So arginine's in our body. It's a normal amino acid. It's not toxic or anything. And it was given in 3 doses, either 3 grams, 6 grams, or 12 grams. And this is the pre-value where we have the arginine before it was given, and then the arginine after it was given, plasma arginine levels. And with different amounts, you say 3, 6, and 12, you can see that it really jacks up the arginine concentration dramatically. And importantly, if we measure this RH-PAT index here with the different amounts, we can show that it will go up. They improve the vasodilation with increasing arginine levels. It's sort of a dose response. So the arginine is reversing this abnormality in endothelial function. And if we looked at that over time as the patients were treated, this was once again published by Dr. Yo with us, the RH-PAT was low initially, and with treatment and resolution of the malaria, and the treatment was with typical antimalarials, it returned to normal. Likewise, arginine levels came up to normal. And lactate, which is an indicator of disease, you have hyperlactemia, lactic acidosis with malaria, it normalized. So in summary, this endothelial dysfunction is pretty much universal and common in adults. We've tried to do this in children. We've had some trouble with the children because many of the children we work with are so small that their fingers don't fit into the apparatus well, so we're having trouble with that. The endothelial dysfunction, a lot of that I didn't show you, is directly proportional to the parasite load that we can measure well. And plasma arginine is low in these people, and if you give arginine, it's safe. It normalizes the plasma arginine. It reverses the endothelial dysfunction. It increases the amount of nitric oxide production in these people. Once again, this is adults. Now, there are a lot of other things that I'm not going to address today. Other researchers, we haven't done it, but other researchers have been looking at carbon monoxide, which actually acts like nitrogen monoxide. So CO works kind of like NO. And there are some researchers who are looking at possibly administering small amounts, not enough to kill a person, small amounts of carbon dioxide. This has been done in non-human experiments or experiments with non-human animals, but just kind of interesting. This is a review that we wrote about some of these things. It seemed like it was only a year or so ago. I guess it's been 5 years ago.
Is there any data in smokers because of the increased carbon monoxide?
Well, there's a lot of data about carbon monoxide in smokers with reference to forming carboxyhemoglobin. And there, rather than being beneficial in that setting, and that's one of the fears of the toxicity here, is that it reduces the ability of the hemoglobin to unload oxygen, so it can be toxic. But in their mouse experiments, it didn't do much. I don't want to overemphasize that. It's highly experimental right now. But you're right. People who smoke, especially cigar smokers and pipe smokers, get a lot of carbon monoxide. Now, I want to go back to this. I'm not going to describe everything, but I want to emphasize about this, the L-arginine analogs, asymmetric dimethylarginine and symmetric dimethylarginine. This is the asymmetric dimethylarginine and the symmetric. So here's the regular arginine. These have the methyl groups put on here, and those are formed in our bodies. All of us have some floating around. If you have renal failure, severe renal failure, these can be increased. This is how they were initially discovered. Patients with renal failure, people were looking in the urine and the plasma for something that might be causing high blood pressure. And it was discovered that the ADMA levels were quite high and that they would be inhibiting NOS, therefore inhibiting nitric oxide production, and therefore we get vasoconstriction, hypertension. These arginines are natural in our body. In addition to these two, there's monomethylarginine. This is a compound in this nitric oxide pathway that was really originally identified by Dr. John Hibbs in Salt Lake City. He's looking at tumor cell killing by activated macrophages and inhibits nitric oxide formation. This ADMA and the SDMA are formed from tissues, and the most prominent are probably muscle and maybe red cells, especially if you have hemolytic anemia where there's a high turnover of red cells, high turnover of hemoglobin. You actually get these methylarginines formed, and then you can synthesize them with a specific enzyme called arginine methyltransferase. And once they're formed, like if you have ADMA there, there's a mechanism to get rid of them, like this long name here, dimethylarginine dimethylaminohydrolase, the DDAH. And it's been shown, especially in cardiovascular medicine, that variances in these genes, like polymorphisms here, can influence the production of ADMA or levels and have some relationship. And there's some evidence from the upcoming tropical medicine meetings that this may be important in parasitic diseases too. Now, the ADMA, as I mentioned, is a competitive inhibitor of NOS, and SDMA doesn't inhibit NOS, but it does inhibit the transport of arginine into the cell at fairly high concentrations. We're not sure that's physiologic. It may be so high it's not going to be important. But both of these would serve to limit the amount of nitric oxide production. Other researchers have shown now that the SDMA is very pro-inflammatory. It causes cells to liberate reactive oxygen species like superoxide, and there in itself it can be bad. And importantly, in population studies and with individual human patients, they've been shown, or researchers have shown that SDMA and ADMA levels are independent predictors of mortality in cardiovascular disease like atherosclerosis, coronary disease, and in renal failure. And then, as I'll show you, that's basically what we found in malaria, too. So once again, Dr. Sen and Dr. Anstey led these studies. in Indonesia, where it was found that increased ADMA in severe malaria is associated with low NO production and with mortality. The adults showed that the levels of both of these were increased, and they were independent predictors of nitric oxide production, endothelial dysfunction, disease severity, mortality. Emphasize again mortality. If you had a real high level, it meant that you were very likely to die. And this is a graph showing the levels in the different disease states of the ADMA and the SDMA. And we've done the studies now in the Tanzanian children with malaria in the different categories. And the findings are slightly different. I'm not a pediatrician, but they always keep telling me that children are not little adults, right, Dr. Clemens? They're definitely different, and we're trying to understand that. Here, there's some subtle differences, and I won't go through all the details, but initially when the patients come in, this is arginine levels, so there's the healthy children. Notice they're low on day 0 in the children with malaria, and then gradually the treatment arginine comes up. ADMA is actually not elevated in the children, but over time with treatment it will go up, and then eventually, like day 7 to 14, it will come back down to normal. But the SDMA is elevated, and it stays elevated for maybe 3 to 4 days, and eventually comes down. And then this arginine to ADMA ratio, which is a general ratio that reflects nitric oxide availability, is low for numerous time points. So just the general message here is that these arginine derivatives, the methylarginines, are elevated in malaria, and they appear to influence nitric oxide bioavailability. And we think that they increase oxidative stress, and they may have some importance on maintaining the reduced state, the BH4, the tetrahydrobiopterin, that's so important as a cofactor. They are associated with poor outcomes in cardiovascular functions in adults, but here in adults and children with malaria, that association holds up. I'm going to summarize things and show some of these again. This may be repetitive, but I think at times on these complicated pathways like this, it's useful to look at it. So remember, arginine is converted to nitric oxide citrulline by this enzyme. Also, arginine, instead of going to nitric oxide, where the NO can have influence on other cells like endothelial cells, platelets, macrophages, etc., if there's arginase present, an enzyme that degrades arginine to ornithine and urea, and then later it's disposed of, if there's arginine there, there's a competition, and so you don't get enough arginine to provide a substrate for nitric oxide synthase. Now, we've looked at this in great detail in the children and adults. We've shown that arginine levels are low. We've also shown that some of the precursors to arginine synthesis are low, and I won't show all the data for that, but the data are very impressive. They're very, very low. We've also shown, as I demonstrated today, that there's decreased messenger RNA for nitric oxide synthase, decreased protein, decreased enzyme activity, decreased NO. And then in measurements that we do on plasma and some on cells, we've demonstrated in adults and children with malaria that there's increased arginase in the plasma. We think that comes from one or two places. One is red cells themselves have arginase in it, the arginase 1 isoform. And so if you're lysing on part of this red cell infection, as they pop open and release their merozoites, they also release arginase and hemoglobin. So that could be degrading that to ornithine and then going on to all of these other pathways. But also, monocytes and macrophages activated the right way can produce arginase 1, arginase that could influence this. And then the other parts of this, and I won't go over this too much, I'm not going to show the data today, but we have shown that tetrahydrobiopterin, this critical cofactor, is decreased in patients with severe malaria. And then there are these inhibitors that are present. There's the cell-free hemoglobin. So you see sort of everything's working to cause decreased nitric oxide bioavailability here. And what is it? We're still trying to answer the question, what is it that's causing the low arginine? We don't know whether it's because there's arginase working on these cells, because there's not enough synthesis. So to try to answer that, we've started some studies where we're using isotope-labeled amino acids, specifically, mostly here, L-arginine, arginine. These are non-radioactive isotopes in which they can be volatile. Dr. Millington is here today. He's an expert in that area. He's helping us doing the work. And with this stable isotope-labeled arginine, we use it sort of as a tracer. So we can put that in the patients and then trace where it goes. What is it converted to? Is it converted to nitric oxide derivatives? Is it converted to or NOS derivatives? Is it converted to arginase breakdown products? Is it just not being synthesized, et cetera? So we give this stable isotope-labeled arginine to the children. We started these studies back in August of this year, and it's a big deal to get this done. It's difficult to even do it here at Duke. When we do it in Tanzania, it's more difficult to get all the right approvals from the approving bodies, as you might imagine, but also just the logistics of doing it was difficult. But we give a bolus of this, and then we give IV administration chronically at a very steady level for about 3 hours and draw bloods at multiple time points, and then measure by mass spectrometry to determine the L-arginine origin and fate. This is sort of the protocol. I won't go through this in detail, but we work with normal children, age 4 to 8, and children with severe malaria. And we give first a bolus of the arginine IV, and then a constant infusion at a constant rate, very carefully monitored over 3 hours, and then have multiple blood draws before and during. And then we separate the plasma and process these. And then we also measure all these other things I've been talking about today. This is a picture of the patient's mother. This is actually a healthy control. Gave us permission to take. This is our research unit at the Hubert Kairuki Memorial University Hospital, where the patient or the control is lying here with an IV sort of in both arms, where we have to have one for the infusion and one for the blood draw. And then this is our research nurse, one of the physicians, Dr. I.M., one of the physicians, Dr. Salvatore Florence, lab technician, Loix, and Dr. Makambo again. And this, you can see our infusion pump here. We have a syringe infusion pump where we put the arginine isotope in, and with a very precise infusion, exact amounts over this period at a steady level. And we have all of these samples. We're anxious to analyze them, and we think we'll be able to give a more definitive answer, and not just say, well, it could come from here, it could go to there. We feel like we can nail that down. I hope Dr. Millington's shaking his head yes there. So what I've done is go through a lot of things, and it's now time to say, so what? I mean, how does this make a difference? Will this lead to any better treatment? Will it help in the management of malaria? Can it help prevent malaria? The way I think about it, and I've listed things here, we do have some possible nitric oxide-related adjuvant malaria treatments. It's important to think that what we're thinking about here, if you're going to do something to increase NO, it has to be in addition to the usual antibiotic treatments like artemethers, et cetera. One is you could increase the NOS substrate, like I've talked about, giving arginine or even citrulline. We've done that. We can increase arginine, we increase nitric oxide production. We haven't proven whether it helps the malaria yet, and those studies are going on now. The other is you can inhibit the destruction of arginine by giving an arginase inhibitor. There are some things, like here, these people, not us, but other people have shown that if you give N-hydroxyarginine, a derivative of arginine, it will inhibit arginase activity. It has the benefit of actually itself serving as a substrate for nitric oxide synthase. So it inhibits destruction and it increases production. So that'd be great. You could increase NOS enzyme activity and production with something like cytokines. Remember, so gamma interferon, alpha interferon, LPS, TNF. People have tried this in the past, not with this in mind, but people have given different interferons to patients with malaria. It hasn't been beneficial, and that remains to be seen whether that'll work. We could reduce or somehow inhibit endogenous NOS inhibitors from forming, like this ADMA and SDMA. I think that may be hard to do, but we're thinking about it. We could also decrease nitric oxide consumption. And there, we're mainly talking about hemoglobin release from lysed red cells. And I don't know how to do that other than to adequately treat the malaria. So I don't have a good idea there. And then the other, which may be more promising, would be to provide more of this reduced tarin, the tetrahydrobiopterin instead of the biopterin or dihydrobiopterin, the actual cofactor. These are safe to give. There are some metabolic diseases, mainly children and other adult diseases, where these can be given safely, and they will increase it. And then finally, you could just give NO in some way. There are researchers from different places that have trials underway now where they're using NO gas. So this is used like in our intensive care units, newborn nurseries, et cetera. You can give NO. It is a gas. I think of NO kind of like oxygen. It dissolves in our tissues, but it comes in the bottle as a gas. And then certain NO prodrugs. Nitroglycerin, as I mentioned, is a prodrug for nitric oxide. Some of the long-acting nitrates we use clinically for angina pectoris are this, and then we have some other ideas there. If I had to choose anything here, I would probably choose these that are highlighted in blue to attack, and we're thinking about those and wanting to start studies in those. So I've covered a lot of areas and a lot of ground here. You've been courteous listening to them. I appreciate that. And I'm very happy to try to answer any questions if you have any questions.
Thank you.